Just want to check the volume. I guess it's okay. I wanted to mention a couple things. I have the notes from Chapter 13 for the first two sections posted, and I have the next assignment for Chapter 12 also. I did decide that all of the problems in Chapter 13 will be put together. I did cut out a couple problems, so it's maybe a little bit longer than some of the other assignments, but not a lot longer. So this week, I wanted to say a few more words about Chapter 12, Section 7, but this week I want to spend most of the time working on Chapter 13. I don't know if we'll quite finish it this week, but we'll probably come fairly close. This deals with the analysis of covariance. So I'll be posting the lecture notes for Wednesday, either tonight or tomorrow night. So now what I want to do is go into RStudio. And before I get started in Chapter 13, I want to go back a second to the repeated measures model. This is actually Section 7, so I need to bring that up. Let me knit this so we can sort of see the gist of what we're doing here. Now, I mentioned that we have two types of what I would call hierarchical models, or multi-level models. And they're multi-level in the sense that there are two error terms. We sort of have what's called a whole plot treatment regimen, and we have a split plot or subplot treatment regimen. And in Section 6, we looked at the situation where the whole plots were designed in a completely randomized complete block design. So you had a block design, and then you had a treatment was randomized within the block, and then you had a subtreatment type. So you had two treatments, but it's not strictly a factorial. Although on the surface it may look as such, it's not really a factorial. There are two error terms in terms of the way this is randomized and sampled. Now the difference between 6 and 7 is what you do on the whole plots. On the whole plots, again in Section 6, you did a block design, whereas in 7 you do a completely randomized design. And then typically in Chapter 7 we're looking at the situation where it's repeated measures over time. There are other cases this could arise. And so if you look at this is the coli experiment, or E. coli, and we were looking at whether or not the treatment, which was the LPS, was effective compared with the control in reducing the number of bacteria. And in addition to that, these were done, measurements were taken at 0, 20, 40, and 60 minutes. So that was a repeated measures, and you had different rats sampled within each of the treatment types. So that was the basic design, and the way I had done this was in the AOV. It allows an error term, and so I had modeled it as, and what we were modeling is the effect on the heart rate. And so we're looking at heart rate tilde, and notice I have treatment times. And so that forms a factorial. However, the treatment is the main plot level. And so you have rats sampled within treatment, and then you have repeated measures over time. And so the top level is the treatments, and then rats within treatments, and then measured over time. So we have a time effect, and then we also have, and there are 4 times, so therefore degrees of freedom is 3. And then we have the treatment by time interaction and the residuals. Now, the residual in the top part is actually rats within treatments, and then the subplot residuals is actually that interaction with time. So that's where you get to 12 degrees of freedom for the residual. Is you have, if you think about the residuals in the top, in other words, here, this residual. That residual represents rats within treatment. And so if you think of the interaction, each rat sees each time, and so therefore there's an interaction. You can think of it as a crossing effect between rat and time. So what you have for the residual in the bottom is really the rat by time within treatment, and so that's this. And so therefore the 4 gets multiplied by the 3, which gives you 12 degrees of freedom. So that is the design that we have. And so we can see that there does appear to be an interaction. Usually in these studies, the interaction is the key thing you're interested in, because it's often the case that there is an interaction. You can see that, in particular, there appears to be some interaction, and you can see from up above it is significant. And so you can see that's mainly this crossing effect going from time 0 to time 20. The control starts higher and then drops more, whereas the treated drops more slowly, and then they drop off at about the same speed. So that the model is alpha sub I plus beta sub ij, and that the beta sub ij is the rat within treatment effect, and then the gamma sub k, which is the factor C, or the subunit treatment. And generally speaking, both the whole plot treatment and the repeated measures treatment, or the subplot treatment, those are generally fixed. On the other hand, the rat, which is the beta, is a random effect along with the epsilon, which is the residual variability and among the subplots. So this is the expected mean squares, and you can see that the experimental units within factor A test the A factor. In our case, that was the treatment tested by the rats within treatment, and then the residual tests the other 2. And so you have 2 tests. Now, in terms of multiple comparisons, I went through this some last time, and that's why I'm not spending quite so much time now. In terms of, I did, didn't I, go through this? I thought I did. So, based on this expected mean squares, and again here, the alphas and the gammas are the fixed effects, we can see how to test. And again, that sigma squared sub b is actually the rat variability within treatments. We call it B, but it's really the rat variability within treatments. And so it's clear how to test. Factor A is tested by the experimental units, and then the subunits test the, in our case, the time and the treatment by time. So when you look at the analysis of variance, by the fact that I put an error term in here, treatment by, I actually put it as treatment by rat as the error term. That will break out these two levels. In other words, it will break out the treatment by rat error term, and in addition, you'll have the error term from the residual variation. So that's the way we do our model. And here is the model. And the expected mean squares give us insight into how to test. Now these tests above are done correctly. That is, if you look at the test, they're done correctly. And I'm going to show you in JUMP how to do this also. Now, if we were going to do multiple comparisons, we could do two overall A means, which in our case is comparing the treatments. Of course, we only have 2 treatments, so we don't really need to do multiple comparisons in our case. And so there we're going to be looking at, let me blow this up a little bit so we can see it better. There we're going to be using the mean square for B, and that mean square for B is estimated, again from the output, it's estimated as 1206. We're going to use that as the error term for making comparisons among A. So that's the standard error will be 2 times that mean square divided by B times C. And B and C is what you're averaging over. When you're looking at the A means, you're averaging over the 2 other treatment levels. And so what I'm doing, if I'm comparing 2 overall C means, I would use the other error term, which is 2 times the mean square for the subplot remainder. And again, since I'm comparing C means, I'm dividing by A and B. Now you might also want to compare 2 C means at the same level of A, for example. So if you look at the plot, you might compare times at the same level of A, or you might want to compare, in other words, we might want to compare, the other thing that isn't given in the book is you might want to compare the 2 A means at the same level of C. I don't think that's given in the book. So at any rate, this allows us to make those appropriate comparisons. Okay? So usually if we reject the hypothesis, you'd want to do follow-ups. I probably should add a few things in the notes on some of this. But of course, there are only 2 A means. So therefore, that's a fairly straightforward comparison. Since there are only 2 A means, that is, there's only the LPS and the control. So if you say that the A effect is significant, you're saying there is a significant difference between those 2. Okay. So that's the gist of the repeated measures. Now what I'd like to do is to show you how this model would be done in JUMP. So I need to go in and get the data, so I'm going to connect to the server. Hopefully they'll be here. Chapter 12, Section 7. And here's the data in JUMP. That's what the data looks like. So you can see that there are 2 treatments, LPS and control, and there are 3 rats within each treatment. And then there are 4 times that you measure each rat. and then these are the heart rates. And so we want to know, is the heart rate less for the controls versus the treated, and does that depend on the time? So that's the basic model. So let's go in and take a look at this. So if I go in and fit model, my y variable is the is heart rate, and then I have treatment, and then I'm going to look at rat within treatment. So I'm going to nest that. So that's an indication that's rat within treatment. And then I have time, and then I have treatment. I have treatment by time. I'm going to cross that. Now, that's all I have to put in the model, because the residual will come out as the residual. So I don't need to put the rat by time within treatment interaction. I could, but then it would have 0 degrees of freedom with residual. Now, what we're going to do is make this a random effect. Okay. And then we'll do the expected mean squares traditional. So this is the basic model that we're running using jump. And so I'm going to go ahead and run this. Okay. Perhaps I should have done a minimal report, but there is a reason that I didn't. Let me just look at this part for a moment. Now, first of all, if you look at the treatment, the rat within treatment, you can see the rat within treatment. Oh yeah, they're actually forming a synthetic test. Let's see. Yeah, the treatment, they're using the rat within treatment to test, and the residual to test the time and the treatment by time. By the way, if you want to know the rat-to-rat variability, you can also test the rat-to-rat variability with the subplot residual effect. And so, but we're not particularly interested in this last one. What we're interested in is the treatment, the time, and the treatment by time. And most of the focus is on actually the treatment by time. And you can see that the treatment by time is highly significant. In fact, you can see that both the time and the treatment by time. However, you can see that the treatment effect is not really significant. Now, if we compare this, I don't know where I have this. If we compare this, the 4.26 and the 4.26 here agree. The time effect is 2676, and that's what we have for the F value here. And then 184 corresponds to this 184. So you can see that that everything agrees between jump and what we're doing with Harner. So this is how we do it. Now, if you sort of expand this out a little bit, let's go up to the top, and we see the treatment by time. Now, I think we can get the interaction by doing the least squares means plot. Yeah, by doing the least squares means plot, you can see that plot, and you can make this bigger if you pull on there. You can see that looks very familiar, right? That's the same plot we had. And right here, they give the least squares means, and they give the standard errors of the least squares means. And so that's the least squares means are actually what are plotted here. Now the least squares means are going to be the same as the sample means, and because it's a balanced design, there's no missing data. But at any rate, this is what we call the interaction plot. And if we wanted to make inferences, now the problem is in JUMP, it's not totally clear to me how I can make inferences, because if I go up here, it lets me do students T, okay? But it comes out with all the possibilities. In other words, it comes out with the 8 different, it puts the 8 combinations of treatments, the 2 treatments times the 4 times, it puts them together into, it puts them together all together. And the problem is the error term it's using will be the residual to do this, and that's not necessarily, depending on the contrast you're looking at, that's not necessarily the correct error term. So I don't quite know in JUMP if you can easily get, if I wanted to compare, for example, the treatment, the LPS to the control within a given time. So if you look at this plot, suppose I wanted to compare the treatments at the same level of time, then that's not really correct as it's done over here. You could say, for example, you could see right here. I need to expand this up, I guess. You can see, for example, let's see, control 0, and then so if you look at this cell right here, this is comparing the LPS 0 to the control 0. So here's control 0, and here's LPS 0. So if you look at that cell, you would think that you would be comparing the LPS with the control at time 0. The problem is that the error term they use for this is not correct. And so that's a little bit of a problem in JUMP, and I'm not quite sure how you can get around it. In R, of course, you can program it to make it happen. In JUMP, I'm not sure there's any way you can correct the problem. They do give the if I look here are the least squares means, and I can compare the contrast or the least squares means students T. But if you notice, this test is actually going to be the same as the whole plot test because there are only 2 treatments. If there were more than 2, then this would be how you would do multiple comparisons for the whole plot. In other words, the whole plot treatment is this effect right here. And if I had 3 levels, then I could do multiple comparisons. So I could compare level 1 with level 2, and level 1 with level 3, and level 2 with level 3 for the factor A, which in our case is the treatment. But in this case, we only have 2 treatments, and therefore there's only 1 comparison. And so that 1 comparison corresponds to this single degree of freedom over here. So if I look here, the treatment, where's the table? Well, the treatment only has 1 degree of freedom, is what I'm saying. Yeah. So the degrees of freedom in the numerator is only 1. So that is the contrast of comparing LDS to control. So at any rate, the results in JUMP are the same, but as I said, you could do it, but you'd have to do it, I guess, by calculator or by hand, do the multiple comparisons that are most meaningful. So that's the way you do it in JUMP. And JUMP has some advantages. It's very easy to do. On the other hand, if you can't do it, you have sort of no way of getting around the fact that you can't do it. You have to do it by hand to finish out the analyses. Are there any questions on that? Okay, well, what I want to do now, so you have an assignment, and basically I have about 1 problem for each of the 7 sections in chapter 12. So I think there was seven problems, which is a little bit longer, I think, than your other assignments, which were 5 or 6 problems per assignment. So it's slightly longer. Any questions? Okay, what I'd like to do now is to go in and look at the first 2 sections of chapter 13. And I'll do it in jump also. So let's go to chapter 13. And I've passed out what I call C13S1 for that's section 1. It actually has section 1 and 2 in it, but it gets awkward to label it as such. So let me set the working directory. And then I'm going to nip this. So what is the analysis of covariance, and why might you want to do it? Okay. Well, think back to chapter 11, where you had the one-way analysis. In chapters 10 and 11, we talked about the one-way analysis of variance. Well, suppose you added, there you had an outcome variable which was continuous. You have a predictor in chapter 11, which is categorical or a factor. But suppose we want another predictor that's continuous. And so chapter 9 talked about linear regression. So suppose you kind of combine the idea in chapter 9, which has a continuous predictor, so y tilde x, where x in chapter 9 was continuous or numeric, and in chapters 10 and 11, the x was a factor. So we did the one-way analysis of variance in chapters 10 and 11. Now suppose you want to have 2 terms in your model. Now we've done this before when we did the two-way analysis of variance and so forth, where we had 2 or more factors. And then we started looking at issues. Are some of those factors random, or are they fixed? So we expanded the idea of a single numeric predictor or a single factor as a predictor to the 2-way and the 3-way and various designs, which all are based upon factors, either 2 factors or 3 factors, or designed experiments that are multi-level, but again, factors. And in some cases, those factors can be random as opposed to fixed. But now we're going to go a slightly different way, where we're going to say we have a factor, but in addition to that, we're now going to have a numeric variable. Now I should mention that these ideas can be extended so that you can have multiple numeric variables, or in fact, multiple factors. And if you get to the point of having covariance with hierarchical models, the covariance, which are going to be numeric, can be measured at either the whole plot or the subplot level, or the subsample level. So this idea of how you integrate these things together is something you need to think about. I mentioned before, when you think about statistics and you think about a study, there are really 3 aspects. There's a sampling design, there's an experimental design, and there's a treatment design. The treatment design deals with how the treatments relate to each other. Are they nested? Are they crossed? But it also relates to things like, are they fixed or random? The experimental design is how do you control variation? And the way we control variation, those sources of variability that we use to control the variation are usually random components. But we also can have both factors or categorical variables mixed in with numeric variables. So we're going in this, what we're going to do here is to mix in, we're going to take the one-way analysis of variance and add a numeric variable to it. So on the experimental units, which are classified in some way, which form the factor, we're also going to do a quantitative measurement. Now, why might you want to do this? Well, there are a couple of reasons. You might want to increase the precision of your study. So suppose Y is the pound lost under 3 diets. But Y may also be related to X. So you have diets, and we want to know if the diets are different. But Y might also be related to X, which is the pounds over rate at the start of the study. So how much weight you lose may be related to how heavy you are. So a person that's way overweight has a lot more that they potentially could lose than someone who's just a little bit overweight. So we do have to be careful that we randomize the people to the diet, because we don't want all of one diet to get the really heavy people and another diet to get the very light people. So what we want to do is we want to randomize the people to the different diets so that there's equal chances of a particular heavy person getting in any one of the treatments. That is one of the diets. So this allows us to, the within treatment variability can be reduced by removing the regression of Y on X from each group. I'm going to show you what I mean by that. We can also control for extraneous variables. Suppose Y is a teacher's salary, and you have three school system, and X is the educational level of the teacher. So you would think that the educational level of the teacher would also affect what the salary will be. So again, we like to control for that. Again, we don't want the differences to be just because there are differences among the teachers. We want to try to control, at least for the educational level of the instructors. And then we may just want to compare the regressions within groups. So if Y is blood pressure measured on different racial groups and X is the salt intake, you might want to look at the regression of blood pressure on salt, but it might depend upon the racial group. So if you have more salt, then this may make the blood pressure go up, but the blood pressure may go up differentially depending on the group. So you don't want to fit just a single regression in this case. We want to actually fit a regression within each of the treatments. But we don't do 3 separate regressions. We do it all in one model. So what is the model? Well, the basic model that we're looking at is Y sub ij is mu plus alpha sub I plus beta. Now here comes the regression coefficient. Beta of x sub ij minus x bar. Now x bar is the grand mean plus the error term. So you can see that we have two things here that in terms of the predictor. We have alpha sub I, excuse me, which is the treatment effect. And generally speaking, that's going to be fixed. And then we have the beta, which is the common slope. If there are a treatment effects, little a, then we have beta as the common slope. Now you might ask, well, suppose the slope depends on the treatment. Well, that's a model we're going to look at, and the only difference is we put a subscript beta sub I. In other words, I should add that into this because later on we actually do look at that model, and that's actually not in the notes I've given out so far, so I may add that because the model changes the mu plus alpha sub I plus beta sub I, and beta sub I would say that the slope depends upon I, which means it depends upon the treatment level. Is that clear? So we're not going to look at that today, but we'll look at that. We'll look at that aspect. We'll look at that aspect on Wednesday. So a lot of, if you look at the book, because the book was taking the point of view that you should be able to compute all these by hand, it in a way kind of limits what you can look at. And of course, when you're using R, we don't have those limitations. Now, we're going to assume that X is the continuous or numeric variable. It's the predictive variable along with categorical or factor. And we also have epsilon, as usual, as the random effects error term. And again, we have the standard assumptions. Independently normally distributed mean zero invariance sigma squared. So that's the basic model. And this model can be extended in various ways. As I said, the beta could be changed to a beta sub I, which means the regression slope depends upon the group. But it's also possible to have more than one numeric predictor. That is, we could have more than one numeric variable. It's also possible to have more than one factor. So you can have more than one factor. In other words, the alpha, the treatment could actually be in a factorial, where we have A, B, and A colon B. So we could have a factorial design with a covariate. And we could have multiple covariates measured on each experimental unit. In a multi-level experiment, like we just talked about, we could have a multi-level experiment, and we could have one or more covariates, and they could be measured at the whole plot level or the subplot level. So these are all extensions to the basic models we're learning. Towards the end of the semester, I might show you, just with computer code, how you might do some slightly more complex models than what we're doing today. So then are questions on the basic model. The book spends quite a bit of time in going through how you would calculate this by hand. From my point of view, this is going to be purged. And the reason is that this is what it is for a single covariate, meaning a single numeric predictor. And what you have to do, if you were doing your one-way analysis of variance, you would be doing this last column where it says for y. These are the calculations you did. I've slightly modified it to indicate that this is the y term, but this is what you had to do for the one-way analysis of variance in chapters 10 and 11, that last column. But when you add a single covariate, I have to do the same thing, basically, for x and for the product of x and y. So for every observation, I have to multiply x times y and get a new variable, xy. And I have to do the same type of breakout in terms of this. And there we call this, in other words, here we call these sums of squares, and here we call them also the sum of squares for the x's. And in the middle, we call it a sum of cross products because we're multiplying the x's times the y. But we do the same type of operations. We get the same type of totals, but it's very tedious to do this, and it's fairly tedious to do this, even if you have a single factor and a single covariate. It's not just those calculations, but in this next paragraph or so, I'm sort of go through very briefly what the calculations are in order to be able to test the appropriate hypothesis we want to test. Now the question is, if you were doing it by hand, you would have no choice but to do it this way. Now, how could we upset, almost immediately I could say, if we do it by hand. But this also depends upon a couple things. It depends on we don't have unequal observations. The n's are all the same within each group. If you don't have equal observations, then these calculations become harder. If you have missing data, it becomes harder. And certainly if you have more than one numeric variable, again, it becomes much harder. So fortunately, that is not the case with using the computer. So there's a lot of the assignments that I will be giving in chapter 13 will not have you compute these by hand. I'll give you assignments related to using the computer. Now you'll notice in chapter 12 I modified some of the assignments from what's in the book because again, I wanted you to strictly use the computer to solve the problems, not do things by hand. There are a few things you have to do by hand, but they're very easy. Because I gave all assigned problems I gave, I believe all of them you use R. And that will be the same thing here. So what I'd like to do is to look at a particular study, and let me see if I can find the so if we look at the book, by the way, if you look at a logical organization, putting the analysis of covariance after experimental design is not really the appropriate thing to do. In terms of reorganizing how this maybe should be done, the analysis of covariance should probably come after the one-way analysis of variance. In other words, you go from a simple linear regression where you have a single y and a single x, and then you do where x is numeric. Then you go into two chapters 10 and 11, where you have y numeric, the outcome variable is numeric, and your predictor x is categorical or factor. And then perhaps the next most natural thing to do is to go to the covariate model, where you have a single y, and you have two x's, one of which is factor and one of which is numeric. And then perhaps move to the analysis of variance, because with the analysis of variance, you could also have covariates, which would be a natural extension of those. And also, when you go to experimental designs, as I mentioned, like particularly the hierarchical models, you might have covariates. So the covariates portion should probably move up so that you could then extend the models that you have for the two-way analysis of variance in experimental design to incorporate covariates. Because it's often the case in the models that we have. You have on the right-hand side, that is the predictors, are often both categorical and numerical. It's very, very common. And so if you put covariance up further, closer to the top, after the one-way analysis of variance side, then that teaches you the basic technique of the analysis of covariance and lets you integrate it into more complex designs, either treatment designs like the factorial designs or experimental designs like the hierarchical designs. And so, therefore, it's going to move up. So, I was trying to find this example. You can see the calculations. It doesn't look like fun, does it? I only gave actually part of this. I think I only gave this. But you can see, computing these by hand is not pleasant. And I don't really think it adds very much to it. In fact, it's just tedious. The important thing is to understand the models in which you're testing and so forth. Okay, so here's example 3.1. We have 3 different advertising media used for each of 5 fast food restaurants of a certain franchise. So you have 15 restaurants, and what I was trying to remember is what we're measuring for X. The number of employees at the restaurant is actually what the X is. So if you look back here, you have three different advertising media, and you have employees, and then you're looking at profit. So those are the three outcome variables that we're using. So initially you might say, let's go back. Okay. Initially you might say, okay, well, let's just fit median employees. Now notice that we made median was probably, when you read into data this way, read.table, by default, anything that's character, such as median, should be automatically converted to a factor. But if you want to be sure, you can use the factor function. So median gets factor of median, which makes sure that, in fact, it was converted to a factor. The fact that it's character doesn't mean it's a factor. You can have character data that's not a factor. So you want to make sure that it is a factor. So in other words, in this model, using LM, we say profit is modeled as a function of median plus employees, where median is a factor of employees, which is numeric. So here we have a factor, numeric, and the outcome variable is also numeric. Now, everything we're studying in this course, except at the very end, the outcome variable, actually, if you look at the material earlier in the book, they talk about categorical variables, for example, in contingency tables. And in that case, you could consider, okay, I have two factors, and one of them is an outcome variable, which is a factor, and one of them is a predictor, which is a factor, and you end up with a contingency table, and you can make inferences about that. You can also do that as a model, which is called a log linear model. You don't have to, strictly speaking, if it's only a contingency table, it's probably good enough if there's just one factor and one outcome variable that's also a factor. But in general, we use what are called log linear models to model. If everything is categorical, then we use what are called log linear models to model this. Now, at the end of this book, if we have time, we're going to do an example where the factor is binary and we're trying to predict it. In other words, it's the outcome variable is being predicted, and it's a factor. And this is called logistic regression. So if we have time, we'll do an example of that. Okay, so I want you to note, I did two different models. I did one where I say median plus employees, and I did another one where I do employees plus median. And you'll notice that it's not the same. It's not the same. In other words, look at the sums of squares for those two. They're the same. Look at median in the top and in the bottom. Median has the sum of squares of 310 up here, but down here it's 502. And the same is true for employees. The sum of squares is different for the two. Why is that so? Well, by default, R fits the model sequentially. That is, it will fit, if you think of it will fit it in the order they're listed. So it will fit median, and then it will add employees to that model. So median fit without employees in the model is what's done at the top, but in the bottom, median is fit after employees is in the model. And there's a big difference. So in the top, when we fit median, it's as if we don't have the covariance. It's not there. So we get a sum of squares due to median. But covariance not in the model. So we're not adjusting for the covariance. Whereas in the bottom, when we fit median, we're adjusting for the covariance. The same thing is true of employees. In the top model, employees is adjusted for median. But in the bottom model, employees is not adjusted for median because it's the only thing in the model. This is called a type 1 sum of squares. Now, if you look, so the point is, in order to get the correct test, we typically want we want the term in the model after adjusting for the other terms in the model. So in the top one would be where we would test for employees. So this would be the test up here for the employees. But it wouldn't be the test we would use for median because, in other words, if we said that the X is not related to the outcome, in other words, if we fail to reject the null hypothesis and conclude there is no employee effect on the outcome, and we drop it from the model, then it would be okay to look at the median without employee in the model. But if in this case, we say the employee is significant, therefore it needs to be in the model. And it's significant after median is in the model. So if we go down below, that's where we're going to test median. We test median after adjusting for employee. And again, that is significant also. So in order to get the two tests, we had to run it twice. Now, if you run the ANOVA, which is called a type 2 test. Let me back up a minute. Now, R does not, in the base package or the statistics base, have any way to combine those into one table. Now, SAS introduced the notations back literally around 1970, what was called a type 1, type 2, type 3, and type 4 models or sums of squares. The type 1 fits sequentially. So if I have A plus B plus C in my model, it fits A by itself. Then it fits B after A, given that A is also in the model. Then it fits C after B and A. So it sequentially fits them. But usually if I do that, my test for C would be right, but not my test for B and A usually, often. So the type 2 sums of squares, corrects it in the following sense. If I have things at the same level, let's take a factorial experiment. A factorial experiment would be like A plus B plus A colon B for the interaction. Now that's a two-level model. We have the main effects, which I'll call level 1, and we have A colon B, which is level 2. Now a lot of times you want to test hierarchically. So I would like to test the interaction first, adjusting for A and B. So the interaction would be tested first, and if it's not significant, I may drop it out and just look at the test of A and B. But what type 2 does, it would adjust the interaction for A and B, but it would just adjust at the same level. So it would adjust A for B, but not A colon B, and it would adjust B for A, but not A colon B. That's type 2. So type 1 is sequential. So if I had type 1, let's say I have A plus B plus A colon B. So what would type 1 do? It would test B by itself. Then it would, A by itself. Then it would test B adjusting for A. Then it would test A colon B adjusting for A and B. That's what type 1 would do. What does type 2 do? Type 2 would test A adjusted for B, even though B is after A, but not adjusted for A colon B. It would test B adjusted for A, but not A colon B. And then, because it always tests at the same level, anything above it, so the interaction, would not be adjusted for. So it adjusts for things at the same level. So if you go up to the two-way interactions now, it adjusts for any two-way interactions, but it doesn't adjust for three-way and higher interactions. So if I had A plus B plus C plus A colon B plus A colon C plus B colon C plus A colon B colon C, the two-way interactions would adjust for themselves and everything below it, but not anything above it. That's the type 2. And that's sort of a hierarchical approach to doing things. So what this is doing, now type 3 adjusts everything for everything. No matter, so it would adjust the one way for all the two ways and all the three ways. It would adjust for everything in the model. It always sort of it's always as if it's last within a sequential model. Every term, it's always as if each term is last in the model. Now, what's the difference between type 3 and type 4? Well, not a whole lot unless you have missing cells. What do I mean by a missing cell? Suppose you have a factorial a by b. And you can think of that as a rectangle with cells, right? So if it was a 2 by 3 design, I would have 2 rows and 3 columns. I would have 6 cells, right? And n sub ij is the number of observations in cell I, row I, column j. But suppose some of the n sub ij's are 0. You have no, if it's an observational study, I might have n sub ij would be 0 for some of the cells. And if that is the case, that creates a problem. And to deal with that problem is the main difference between type 3 and type 4 sums of squares. Okay? If you have too many zeros, the study becomes what's called disconnected. And there you have big problems. But you have actually problems even if it's not disconnected. The missing cell problem is one of those problems that is, you have to sort of say, well, what does this exactly mean now? Because I have this whole cell missing. So if you look at that interaction plot, okay, the one we looked at just earlier, if you look at that plot, one of those dots is totally missing. Or maybe more than one. So, at any rate, notice there's a library called car, C-A-R, and in it is a function called ANOVA. Now, there's a function, ANOVA, that's defined in the regular base R. But do you see anything strange about that ANOVA? There's a capital A. That is different than ANOVA with a small a. It is completely different. What ANOVA will do is let you do types 2 and 3 sums of squares for lots of models. The LM function, the GLM function, multi-level models, etc., etc. So the ANOVA with a capital A will let you do it will let you take those 2 tables above and basically combine them into one table below. Look, compare the table below. So look at the 1481 for employees. Well, where does that go? It's not this one. It's coming from here. And look at the 502 for medium. Where does it come from? It doesn't come from here. It comes from this model. In other words, it's doing the one that's last each time. With the correct sums of squares. So, and also notice that it even tells you that it's doing type 2 tests. Okay? So by default, that's what it does. If you want type 3, you have to tell it to do that. So these are the correct tests. I didn't have to run it twice. There's certain people in the R community that I think are a little stubborn because one of whom is Doug Bates, because he doesn't, he's doing a lot of the works with mixed models and so forth, and he doesn't want to hear about this. But in reality, just about any practicing statistician would do it this way. Or let's put it this way, why would I want to run a bunch of different models or do a bunch of different, I'm actually running only one model, but in other words, I'm applying that to media fit. But it wouldn't have mattered. Media fit, I did twice, right? I did it here, and I did it here. I called it the same thing. And it really doesn't matter which one I use. The order doesn't matter. I fit the model. The design matrix does not change. Did I print out the design matrix? Yeah, this is the design matrix. That doesn't change. It doesn't matter the particular order I put it in. That does not change. So the LM function does not change. It's when you put that into an AOV table that's where the difference is. When I do ANOVA, notice the ANOVA here with the small a. which will do type 1 sums. It will do type 1 test, type 1 sums of squares. So ANOVA with a small a does type 1. By default, ANOVA with capital A will do type 2. But it can also do type 3. Okay, so this gives us the correct test, and we can do it. We can take the fit from the LM model and do ANOVA with capital A and get the correct test. Now, by the way, this is, I mentioned this before, but this is the design matrix. That's what we call the X matrix. And notice the first column is for the intercept. We have 4 parameters. What do we have 4? Well, we've got the intercept. You know, if you go back to the model, we have the intercept, which is mu, and alpha sub I, but there are 2 of these. Why only 2? Well, it depends. There are a minus 1 because we have the constraint the sum of the alpha sub I is equal to 0. And then we have the regression coefficient. So what's that? As far as R is looking at what are the predictors. Well, these are the predictors. The intercept is the coefficient for mu. And then the beta, if I think of that as a matrix, it's a 15 by 4 matrix. And if I multiply the vector consisting of mu, beta, alpha 1 and alpha 2, then if I multiply those times these, once I estimate these parameters, I get the predicted values. So this represents the intercept term. These are the actual x values for the covariate. And then these are the dummy variables. Here I have a 1, 0. That says alpha, for these 5 observations, I have an alpha 1. I don't have an alpha 2. And there is no alpha 3. Because alpha 3 is equal to minus, remember the sum of the alpha sub I is equal to 0, right? And so alpha 3 is minus alpha 1 minus alpha 2. So the first 5 observations, for the first observation, I would have mu plus 10 times beta plus alpha 1. That's the first observation, plus the error term. Then for the second observation, it's mu plus 14 times beta plus alpha 1 plus the error term, and so forth. Now when I get down to observation 6, I have mu plus beta times 21. I'm assuming a common slope, plus alpha 2. Notice the median 2 up here. For these 5 observations, they're all like that. Mu plus beta times the x plus alpha 2. So what is it for the third group? It's mu plus, let's take observation 11. It's mu plus beta times 34 minus alpha 1 minus alpha 2. That's because of the constraint, the sum of the alpha sub I is equal to 0. Suppose you would have said this, just so you understand a little bit better why I need to have the sum of the alpha sub I constraint on there. Suppose these 1s would be 0s. Suppose these were 0s, and these were 0s here. And suppose I had another column called medium 3. And so what would I have in medium 3? I would have 0s here, 0s here, and 1s in this last group. And then you could say it's mu plus beta times the x plus, in the third case, plus alpha 3. Does anyone see a problem with that? In other words, suppose these 5 observations were 0, these 5 observations were 0 here, and then I had 0s, 0s, and 1s. Does anyone see a problem? If that were the case, the last three columns would add up to the first column. You follow what I'm saying? The last three columns, that is a singularity. That is what we call a singularity. You could not solve this problem with a singularity. So it's because of this singularity that we have to put the constraint on. Some constraint. R, by default, puts alpha 1 equals 0. Fine. SAS, by default, puts the last alpha equals 0. That's fine. Jump does the sum of the alpha sub i's equals 0. So why does mine look like jump? Mine looks like jump because I changed it. In R, I did that. I said I want you to use the sum contrast, which is not the default. That contrast polynomial would be what I would use if I had an ordered factor. If I had an ordered factor, I would use an orthogonal polynomial as the contrast. So we're not using that second constraint, but in all cases we're going to be using the sum. So that's why we have these minus ones, because I said to use the sum contrast, which is the same. Basically, I did this so it would agree with jump, since I'm illustrating jump along with this. So I'm making what we're doing correspond to what jump is doing. If I wanted to make it correspond to SAS, what would I do? I would set the last alpha to 0. There is an option, I forget what it's called, SAS.contrast or something like that. There's an option that lets you directly match SAS in R. So in R, you can easily match jump or SAS by how you choose the constraint. Now this is a little bit confusing, but unfortunately, if you don't put the constraint on, you have a singularity and you can't solve for the estimates. Now you may say, does it make a difference as to what constraint? No, it doesn't make a difference. The coefficients look different, but the predictors and the test you do and everything you do not. The things you do, making comparisons, making multiple comparisons, testing main effects, and so forth, they don't differ. I'll show you what does differ. But where it differs, only it changes your interpretation of what you're looking at, but it really doesn't change anything it doesn't change anything that's meaningful in terms of doing the comparisons you're going to want to do. So it really doesn't matter the constraint. Now, so this is the design, what we call the design matrix or the X matrix. If you think of it in matrix notation, we actually set the problem up, and the way everything is done internally is done with matrix algebra. And we'll be getting into, this class doesn't actually assume you have a matrix algebra course. And so what we're going to do as we go along here, we're going to sort of introduce a little bit of matrix algebra gradually. Fortunately, R is going to do the heavy lifting for you. You don't actually have to do the matrix algebra you don't have to do those manipulations yourself. Now, if I do a summary of media fit, that will give the estimates for the four, the mu, in other words, looking here, this is the estimate for mu, right here. This is the estimate for beta. This is the estimate for alpha 1. And this is the estimate for alpha 2. And then alpha 3 is minus those two above, those two. So I can get, once I get alpha 1 and alpha 2, since they sum to 0, I can get alpha 3, right? So this gives the model in terms of estimating the parameters from this, because what I'm really doing is getting that using LM. And this is a summary, it gives the estimates and their standard errors. So you might ask, well, I'd like to be able to plot the points with the different groups identified, and I'd like to plot the 3 regression lines, which in this case have the same slope. I forced them to have the same slope, but there are still 3 lines for the 3 groups. So I have a regression in each group, but I have the same slope. So that's the model. So you might ask, well, where do I get the regression coefficients? So there's a lot of issues here we have to look at. That little plot, this little plot has a lot of information in it. So, for example, these three that red one is the sample mean. These three red ones are the sample means. The blue ones here, and there's actually a blue one here too, and that blue one, are what are called the adjusted means, or the model means for the covariance model. And notice that the adjusted means are adjusted in such a way that they're all measured at the same number of employees, which is the grand mean. There are 31 employees altogether, I believe. In other words, the grand mean is 31. So we're adjusting the means to the x value of 31. So in effect, this mean slides up here, the regression line to here. This one's right on target, and then this one slides down here to here. So you can see the means were adjusted quite a bit by the regression. But let me come back to that for a moment. You know, I've talked about the least squares means and the model means and so forth. So how do we compute these in R? How do we compute these in R? I actually explicitly did this so you can see. So I want to show you how we got the plot, because this is important. If you don't understand the plot, you do not understand the analysis of covariance, because that plot, in essence, is the gist of the analysis of covariance. At least the simplest models where the slopes are all the same. We're going to do this again on Wednesday, where the slopes are not all the same. So, first of all, I plot employees versus profit. But I did something here. I said, this is the point character. I want the three groups to have three different characters. I don't want just a bunch. I don't want 15 points. I want to know which 5 are which group. And so I said PCH is point character equals as numeric a median. What that does is it converts median to a numeric code, and then it assigns a plot symbol based upon that numeric value. And since there are 3 different values, it will get 3 different plot symbols. Okay, so after that one little statement, what would our plot have? What our plot would have would be no lines whatsoever in the middle of that plot. It would just be, and no color. In fact, the colored points wouldn't be there. The colored points would not be there. It would only be the black symbols. But again, you notice we have plus signs, we have triangles, and we have circles. The circles are clearly associated with this bottom line, the triangles with this line, and then the top line would be the pluses. Now notice the pluses tend to have higher number of employees, whereas, so presumably what we're talking about, this group right here, that has fewer employees. The triangles are sort of in the middle, and then the pluses would be larger number of employees, because if you just look at the values for the pluses and so forth. So at any rate, so at this point I would only have the 15 black symbols, but I would know, I could identify the 3 different groups. Okay, so that's all I have. Now I'm going to draw a vertical line at the mean of employees. Okay, this is the grand mean. This is x bar dot dot up in my model. It is the grand mean of all employees. I'm simply going to get the mean, and I'm going to draw, V means vertical, I'm going to draw a vertical line. That's this line right here. Okay, so I'm going to adjust the means for profit to a common x bar. Now, how do I get the lines? Well, you notice I have AB line, An AB line gives the intercept. An AB line is a line. AB meaning A and B would be the intercept and slope. So you can think of A as being the intercept and B as being the slope. The first argument is the intercept, and the second argument is the slope. So notice that all three of these have a slope of a minus 1.592. They all have the same slope because we're assuming a common slope model. Where did I find that? Well, I found it right up here for employees. That is the slope. So how do I get group 1, which is median 1? I get median 1 is mu plus alpha 1, which is 55.78, minus the 13.1. So this plus this is the mu plus alpha 1, and that will be the intercept for the first group, or the median 1 group. Okay, so that's going to be the line. That's going to be that's actually going to be the one. That's going to be the intercept corresponding to the intercept. There is what, about 42. Okay, so that's actually going to be this group down here. Now this is 10, not 0. So in other words, to get back to 0, you sort of keep going up. Okay, so that's going to be about 42. So this is median 1, this lower group. Okay, now the next group is going to be mu plus alpha 2. So it's 55 plus 1, so it's about 56 or 7. So that's the next group. So I have 55.78 plus 1. That's the next group. And so that actually will be the second group. And that has an intercept of about 56. And then the final group will be mu minus alpha 1 minus alpha 2, right? So minus 13 is a plus 13, and minus a minus 1 is, a minus plus 1 is minus 1. So I have mu minus alpha 1 minus alpha 2, and that will be the intercept for the third group with the same slope. So that is how I now have added this. So at this point, I have now added the 3 lines. Now I still don't have any of those colored points up there yet. Those are the means that I'm going to compute. So those are the 3 lines. So now we have a lot of the information we need. But it would be useful to know where are the unadjusted means, that is, the means unadjusted for the covariance, and where are the adjusted means, the means adjusted for the covariance. Now it might be, notice on the y-axis, these reds, this is the unadjusted mean for the first group, this is the unadjusted mean for the second group, and this is the unadjusted means for the third group. Now these blue, here, here, and here are the adjusted. Notice that the adjusted means are further apart than the unadjusted means. You see that? I'm claiming that this red right here is also blue. It shows up as red, but it's a red and a blue. The adjusted means are here, here, and here. On the y-axis, they're here, and they're here. The reds are here, here, and here. The reds are much closer together on the y-axis if you project over than the blues, meaning the adjusted means are further apart, which means we're more likely to reject the hypothesis. So how do we get the red and the blue points? How are we doing time-wise? Yeah. So what I'm going to do is I say superimpose the least squares means. That is the model means. The model means mean the means predicted from the model. The sample, if you go in and just compute sample means, as you see, they will be quite different. And a lot of people make this mistake. So, what I have to do, I can use the predict, I can say predict media fit, and then I can put a data frame in here I can put a data frame and it will use the model to predict new observations. Well, what new observations do I want? Okay, the observations I want, I'm going to have to put in the median effect. And the model involves medians, so I have to tell it which medians do I want. Well, I'm going to want three means. I want three means with the three medians. So I have three more observations. I should have printed that out. But at any rate, so what I'm going to do is create a data frame that has three medians, and then I'm going to predict them all at the same x bar, which is the grand mean. In other words, I want all of these predictions for each group. I want the prediction at x bar, the grand mean. Now you might say that's only one number. You might say here I really have a data frame that has two columns and three rows. Okay, what does the first row have? The three rows of median are 1, 2, and 3. Roman number 1, Roman number 2, Roman number 3. Those are the three rows for the first column. And what's the second column? Well, I've only computed one number, which is the grand mean. But it will get replicated 3 times automatically. Or it just does that. It's called vectorized arithmetic. It will automatically use it 3 times to fill it in and make it a data frame. So if you actually, I should have printed that out, but if you print that out, it's a 3x2 data frame with 3 rows, 2 columns. Okay? So it's going to get a predictor for the grand mean. It's going to get the predictor, the grand mean at each of the 3 categories, each of the 3 groups. So these are so that's what I get. I call them least squares, and here are the values. A minus 4.11. Let's take a look at it. What I'm doing is predicting these 3 points. What is the predicted value on the regression lines, in other words? So a minus 4.1, well, that's this point, obviously. It's down here. It's a minus 4.1. And then this one's going to be, what, 10 or 11? And so that's 10. And then the third one is 21. That's this one up here. Okay? So that plots the blue points. In fact, you can tell it's blue because I said points. I'm going to repeat the mean employees 3 times and use the least squares mean. So on here I'm going to plot some additional points on the plot, and I'm going to say color equals blue. So I computed the least squares means, and now I'm going to plot the least squares means, but they're plotted all at this grand mean. That's why they stack up vertically. Now, I need to I want to plot, just so you can see the adjustment, I'm going to now plot just the sample means, not the model means. If you hadn't even run the model, you could do this. Just based on the data frame, you could compute the sample means. But the sample means might be quite misleading, as you will see they are. So I'm going to use the tapply function, and this says tapply employees. I'm going to get the mean for each of the medians for the employees. And then I'm going to do the same thing for profit. Now these are just the sample means. tapply is one of the apply functions, and this is the outcome variable, and this is the grouping variable, and this is the function I want, which in this case is mean. So this says I'm going to get the mean for each median for employees. Here I'm going to get the mean for each median for profit. Then I'm going to plot the employee means versus the profit means, and the color is red, and that gives me these 3 points. Those are the unadjusted means. So the red are the unadjusted means. Think about the y-axis and where they project to. If you project them to the left horizontally, then if you project them, those are the actual values. So if I project this across, this will be the profit sample mean. If I project it down, that's the employee sample mean, and likewise here and here. So going across, that's the sample mean for profit, that's the sample mean for employees. So if you fit a model where you ignored the covariant, and you just did a model that said profit tilde median, you would use the sample. You would use those red means to do the comparison, not the blue means. So the covariate has had quite an effect in terms of the adjustment. And notice that we probably fit the model much better. In terms of early on, we talked about the objectives more than one good. Those three straight lines fit the data quite well. Yeah.
It's already one.
Oh, yeah. Sorry. Okay. Thanks. That's a good place to stop. So we'll go ahead starting here next time. Okay, any questions? So start reading chapter 13. We're going to continue on it next time. Maybe open the door so they can start coming in.